considerations on yeah, of these factors?
Mean antigen presenting markers.
Yeah, and meaning that you are, yeah, you're talking about macrophages, apa semua kan? They are antigen presenting cells. Normally at the beginnings of
For
process.
inflammatory reaction, there is additional inflammatory markers other than cytokines, which is other than TNF alpha. We also measure chemokines and addition molecules, which related to the macrophage activation.
Okay.
The macrophage activation is also it is also caused by a stimulation of a vascular cell adhesion molecule, which is also measured in this study, and intracellular adhesion molecules, and also macrophage chemo-attracted protein 1. But I'm not included in these markers because the market is too much. So that's why I just pick the main inflammatory markers such as TNF alpha and NFKB.
Okay. Alright. I still ada question tapi saya bagi dekat Dr. Yus pula untuk komen. Kalau ada masa saya tanya question lagi. Okay, Dr. Yus Mira, first to you.
Thank you, Doctor.
Thank you, Prof. Richard. Assalamualaikum. My Dr. Ahmad. I just have a few.
Waalaikumsalam, Doctor.
First of all, you choose 0.5 concentration of your B bread, right?
Yes.
5 milligrams per kilogram of body weight. So, how you select the effective dose for your study?
Initially, the 0.5 gram per kg of the B bread is determined based on the dose of honey used in previous study, which is equal to 1 teaspoon. 1 teaspoon into the when calculated using KM factor to the rats equal to 0.5 gram per kg per day. So we also did perform pilot study involving 3 doses of B bread. For example, 0.5 gram per kg per day and 1.0 gram per kg per day and 1.5 gram per kg per day. And based on that pilot study, B bread at 0.5 gram per kg per day is more optimum, give more optimum effect to the obesity parameters in our pilot study.
So you did the pilot study.
So we proceed.
And how about the phytochemical compounds of the B bread for your sample? Did you
Sorry, judges. Sorry to interrupt. We actually have come to the end of our 5 minutes Q&A session.
Okay, thank you.
Yes, really sorry to judges, to Ridwan. I know you have another question to ask.
No,
but because Q&A session is also a learning session. I think in order to be fair to all presenters, we strictly make it 5 minutes only. I know it's a little bit feel
no problem.
ralat lah gitu. Sorry.
Okay, thank you.
Okay.
Thank you.
So well done, our first presenter. So let's have the next presenter, the second presenter with the code M19, Futun Al-Rawashdi, to present her research entitled Thymoquinone inhibits growth of acute myeloid leukemia cells MV4-11 through re-expression of SHP1 and SOCS3 and suppressions of FLT3-ITD. Let's welcome our second presenter.
Assalamu alaikum. My name is Futun Al-Rawashda. I am from School of Biomedicine, Faculty of Health Science at UNISA University. My study title is Thymoquinone inhibits growth of acute myeloid leukemia cells, the MV4-11, through re-expression of SHP1 and SOCS3 and suppression of FLT3 with internal tandem duplication. Epigenetic silencing of tumor suppressor genes by DNA hypermethylation is mediated by DNA methyltransferases and plays an essential role in the pathogenesis of acute myeloid leukemia. SHIP1, SOX1, and SOX3 are tumor suppressor genes that negatively regulate the JAK-STAT signaling pathway. Enhanced re-expression of the tumor suppressor genes through demethylation represents a therapeutic target in several cancers. Thymoquinone is the major natural phytochemical compound of Nigella sativa seeds that has a potential anti-cancer effects against several tumors. In this study, we hypothesized that JAK-STAT negative regulators that lose their tumor suppression function in acute myeloid leukemia due to epigenetic silencing could be re-expressed by thymoquinone treatment, and thymoquinone could inhibit the expression of FLT3 with internal tandem duplication. For materials and methods, MV411 cells harboring the FLT3 with internal tandem duplication mutation were treated with 5.5 micromolar of thymoquinone for 48 hours. The methylation profiling of SHIP1, SOX1, and SOX3 was determined by biosequencing analysis. The expression of SHIP1, SOX1, SOX3, the FLT3 with internal tandem duplication, and the DNA methyltransferase 1, 3A, and 3B genes was performed by real-time qPCR before and after treatment with thymoquinone. Aberrant methylation of tumor suppressor genes is involved in the pathogenesis of several cancers. The expression of the tumor suppressor genes, such as the JAK-STAT negative regulator genes, is silenced due to hypermethylation of the CpG islands in their promoter regions, leading to the malignant transformation of the normal hematopoietic stem cells and the development of leukemia, particularly acute myeloid leukemia. Therefore, the inhibition of DNA methylation and the re-expression of the silenced tumor suppressor genes are important therapeutic targets for acute myeloid leukemia treatment. However, the therapeutic efficiency of the conventional DNA hypomethylating agents are limited. Phytochemical compounds such as curcumin were found to inhibit DNA methylation. Thymoquinone is a phytochemical compound, and few researchers have investigated its potential as a modulator of the DNA methylation process. In this study, we identified DNA methyltransferases associated DNA methylation of the JAK-STAT negative regulator genes as a previously unstudied epigenetic alterations that mediates thymoquinone's anti-leukemic effects in acute myeloid leukemia. In agreement with our results, thymoquinone significantly decreased the expression of DNA methyltransferase 1 and 3A in the primary blast cells from acute myeloid leukemia patients and in ML1, COSAMI1, and MV411 cell lines. Thymoquinone also had decreased the expression of DNA methyltransferase 1, 3A, and 3B in T-cell acute lymphoblastic leukemia cell line Jacquard cells. Here, the graph shows that thymoquinone induces SHIP1 promoter hypomethylation in MV411 cells. The percentage of methylation was decreased from 9.3% in the untreated MV411 cells to 7.5% after thymoquinone treatment. The positive control 5-azacitidine decreased the methylation to 5.2%. Here, the graph shows that thymoquinone induces SOX3 promoter hypomethylation in MV411 cells. The percentage of methylation was 4.7 in the untreated control cells, which was decreased to 2% in thymoquinone-treated cells. The 5-ASA positive control shows a decrease to 0% methylation. Our quantitative results of pyrosequencing analysis showed that thymoquinone has induced hypomethylation of the promoter region of both SHIP1 and SOX3 with a p-value of 0.037 and 0.041 respectively in MV4-11 cells compared to untreated cells. Our data supports previous study that has shown that thymoquinone induced a significant inhibition of the cell growth as a result of the significant reduction of global DNA methylation in MV4-11 cells treated with thymoquinone. In addition, treatment with thymoquinone increased the methylation level of the proximal promoter of TWACE1 oncogene and decreased the TWACE1 messenger RNA level which resulted in inhibition of cancer cell growth in the human breast cancer cell lines and cervical cancer cell lines. This graph shows that SOX1 promoter region was not methylated in MV4-11 cells. The percentage of methylation was 1% in the untreated MV4-11 cells, which was nearly similar in thymoquinone and 5-azacitidine treated cells, which was 0% methylation. Our results provide evidence of unmethylated status of the CpG sites in the promoter region of SOX1 in MV4-11 cells, and hence are not responsible for epigenetic regulation of SOX1 transcription and are not suitable for methylation study. Similar methylation status of SOX1 promoter region was detected in previous studies that did not find methylation in the CpG sites in the promoter region of SOX1 in AML patients. On the other hand, the promoter region of SOX1 was methylated in the relapsed group of AML patients. This graph shows that thymoquinone enhances the re-expression of SHIP1, SOX1, and SOX3 in MV4-11 cells. These results were in agreement with the previous findings in which SHIP1, SOX1, and SOX3 functioned as tumor suppressor genes that was suppressed in leukemia cells and re-expressed after treatment with 5-azacitidine and ketosine in AML cells. As a result of the FLT3 with internal tandem duplication mutation, the MV4-11 cells have a constitutive activation of the FLT3 signaling. The inhibition of FLT3 expression significantly inhibits the JAK-STAT signaling in acute myeloid leukemia, and here we examine the effect of thymoquinone on the expression of this FLT3 with internal tandem duplication mutation in MV4-11 cells, and we found that thymoquinone has decreased significantly the expression of FLT3 with internal tandem duplication by 7.7 folds compared to untreated cells, and similar to our finding, FLT3 signaling was shown to be one of several molecular mechanisms underlining the thymoquinone-induced cancer cell growth inhibition. In conclusion, thymoquinone mediates anti-leukemia actions in acute myeloid leukemia by downregulating the epigenetic regulator genes that lead to a reduction in the DNA methylation levels of SHIP1 and SOX3, and restoration of SHIP1, SOX1, and SOX3 tumor suppressor genes. And thymoquinone inhibits the FLT3 signaling by downregulating FLT3 with internal tandem duplication. We suggest thymoquinone as a potential anti-leukemic and a DNA methylation natural modulator. Thymoquinone could be used for future studies to define other genes that are involved in acute myeloid leukemia pathogenesis and may be epigenetically modified by thymoquinone treatment.
Okay, thank you, presenters. Let's go for, please turn on your microphone and camera for our Q&A session.
Assalamualaikum.
Assalamualaikum, Doctor.
Should I call you Futun or Rashwadi?
Futun.
Futun. Okay. Okay, Futun. Please answer in the brief. Is why Thymoquinone?
What is thymoquinone?
Why Thymoquinone?
Why thymoquinone? Because there is a previous study that shows the inhibitory effect of thymoquinone on leukemic cells, especially in acute myeloid leukemia and leukemia in general. So there is many studies that showed the anti-leukemic effect, but the mechanism of action is not well studied.
I think it's just too long of your answer. I'm expecting that something like that you are working. Is it because of Nigella sativa or whatever it is, the active
And
compound?
yes, it's the major component of Nigella sativa, which is used for a long time.
Is it because of that Nigella sativa?
Not because of Nigella sativa, but because of the previous study that showed the anti-cancer effect, and because it's a phytochemical, and many studies showed that it's okay.
Understand. Think whatever it is, yeah. Because okay, I straight to another question because I would like to pass to Dr. Yuspira after this. Why you do not include the normal blood cell as well? Only
normal.
Ah.
Only untreated cells. Why I didn't include the normal blood cells? I think that's because of the shortage of a grant. Maybe I didn't because I actually using 2 cell lines and the work was too long to include other type of cells.
I think it's such a very good study, something like that. Please consider to put the normal cells as well, because to make it your research higher.
You mean to show the safety of the drug or for what?
Yeah, something like that, the effective whatever. Because
like this.
most of the cancer study they just treat and untreated, because normally
Yes, I was thinking to include the normal cells to see the safety, but maybe I will consider that in my future study.
Thank you very much, Dr. Yusmira. Masa very fast. Mencuburi.
Assalamualaikum, Futan.
Assalamualaikum, Doctor. How are you?
Okay, thanks. Okay, just a quick one. Your sample thymoquinone, did you extract by yourself from the Nigella sativa or actually commercially?
And we have actually bought it from Sigma Aldrich.
And it's natural?
Yeah, so we didn't extract thymoquinone. It is already ready to use.
Okay. And another thing is, in your study, like mentioned by Prof. Elizabeth, you doesn't include normal cells, right? And I also noticed that there is no quality control, right? There is no control drug that you use in order to.
No, there was a positive drug which was 5-acetylene.
Oh, but you mentioned the presentation just now.
Yes, actually, when I presented my biosequencing results, I mentioned the effect of 5-acetylene as a positive contra.
Okay, so I think it's better if you can include that positive control and also your normal cell and negative control, then it will give a clear mechanism for your study.
Okay.
Thank you.
Thank you, Doctor.
Okay, thank you, judges. Well done, presenters.
Macam banyak masa pula.
Oh, ada soalan lagi ke?
Adalah, tadi macam masa pendek sangat.
Tadi banyak. Nak tanya lagi?
Tak ada.
Okay.
Soalan yang banyak lah nak tanya, tapi yalah.
Saya pun suka juga Q&A session. Okay, let's have our third presenters. I would like to call upon Amirul Faiz bin Shamsuddin with the code M27 to present his research entitled Association between APOA5, RSNN, RS 662799 and GATM RS 9806699 with lipid lowering effect of statin among outpatient statin users in hospital USM. Please welcome.
Bismillahirrahmanirrahim. Assalamualaikum and a very good evening I wish to everyone. My name is Amir Al-Faiz bin Shamsuddin and the title of my oral presentation is the association between ApoA5 RS662799 and GATM RS9806699, the lipid lowering effect of statin among outpatient statin users in Hospital University Science Malaysia. Before I start, here is the contents of my presentation. So moving on to the introduction. So what is statin? Statin is commonly used treatment to reduce cholesterol levels. And mechanism of action of statin takes place inside the liver during cholesterol biosynthesis process, in which statin inhibits conversion of HMG-CoA into mevalonate, which is a rate-limiting step in cholesterol biosynthesis. As a result, cholesterol production in the liver is reduced. So how does it affect lipid profiles in plasma? While focusing on atherogenic lipoprotein, statin treatment able to reduce the level of atherogenic lipoprotein, which includes VLDL, IDL, and LDL, due to increased uptake to the liver. While for non-atherogenic lipoprotein, statin treatment able to increase the level of HDL level due to reduced transfer of cholesterol from HDL into atherogenic lipoprotein. While talking about statin efficacy in reducing cholesterol level, mainly LDL level, statin efficacy in reducing cholesterol may vary between individuals, even when compliance was taken into account. So there are several factors influencing statin response and efficacy. One of them is genetic polymorphisms. Now here's the figure just to show that there are several genes that are involved in statin disposition, as well as statin efficacy and statin side effects. Hence, based on candidate gene approach, two SNPs have been selected for this study. One is RS662799, which is located in ApoA5 gene, and another one is RS9806699, which is located in GATM gene. The effect of this SNP towards statin treatment have been listed in the table below. Therefore, the problem arises for our study is that are there any relationships between these selected genetic polymorphisms towards lipid lowering efficacy of statin? And we hypothesized that these selected genetic polymorphisms are associated with lipid lowering effect of statin. Therefore, the aim of our study is to investigate the association between these selected genetic polymorphisms and lipid lowering efficacy of statin among outpatient statin users in Hospital University Science Malaysia. Let's focus on materials and methods for our study. The first part is patients recruitment, in which this is a cross-sectional retrospective study, and total number of 229 hyperlipidemic statin users have been recruited from Hospital University Science Malaysia, with the criteria listed below. This is followed by DNA extraction, in which DNA extraction was performed by using S-gene blood examinate from GenAl Biotechnology, in which the extracted DNA quality and quantity was inspected by using microplate reader and 1% agarose gel electrophoresis. After DNA extraction, SNP genotyping was performed by using polymerase chain reaction restricted fragment length polymorphisms, also known as PCR-RFLP, in which the extracted DNA was amplified in PCR reaction, followed by digestion by using restriction endonucleases in the RFLP reaction. The list of primer sequences and product sizes upon successful DNA amplification in PCR reaction was listed in the table, and also the name of restriction enzyme used for RFLP reaction for each SNPs also listed in the table below. After performing SNP genotyping, statistical analysis of the association between selected SNPs and lipid lowering effect of statin was performed by using SPSS software. in which recursive model was used to group the patients according to the minor allele carriers and non-carrier group. And pairwise comparison in terms of lipid reduction between these two groups was performed by using independent t-test and manual u-test. Now let's move on to the results part of our study. This is a patient's demographic profiles. By focusing on the most bottom part of the table, at the mean lipid level at baseline, you can see that our population had elevated total cholesterol and LDL level above normal range. While for non-atherogenic lipoprotein HDL level, the baseline level was lower than the normal range, while for triglyceride level was borderline to optimum level. The next result is the comparison of genotypic distribution of selected SNPs between our population and the reference population by using chi-square test of independence. The reference population here is referring to East Asian populations data obtained from Ensemble database. Now, focusing on this table at the p-value of each SNPs, you can see that genotypic distribution of ApoA5 RS662799 and GATM RS9806699 were significantly different to the reference population. And now to our main finding, which is the association between selected SNPs and lipid-lowering efficacy of statin. Focusing on our first SNP, which is ApoA5 RS662799, this SNP was significantly associated with HDL and triglyceride levels. Focusing on the figures given, you can see that the minor allele carriers had higher HDL level and lower triglyceride levels, in which both effects can be seen after 7 to 12 months of statin treatment. However, no significant association was observed with other lipid profiles. While for our second SNP, GATM RS9806699 was not significantly associated with any of the lipid profiles, therefore was not shown in this result section. Now let's move on to the discussions part. The first part of discussion is on ApoA5 RS662799 association with statin efficacy. While based on previous study, ApoA5 gene had been associated in decreasing plasma triglyceride levels by reducing the VLDL synthesis in the liver. In our current study, it showed that ApoA5 RS662799 carriers had better response to statin treatment in terms of increasing the non-atherogenic lipoprotein, which is HDL, as well as reducing the atherogenic lipoprotein, which is triglyceride levels. Therefore, our results suggest that the minor allele ApoA5 RS662799 may have exerted a protective role against cardiovascular disease risk. And the second discussion is on our second SNP, GATM RS9806699 association with statin efficacy. While based on previous study, this SNP had been associated with protective effect in reducing statin-induced myopathy, in which statin-induced myopathy is well known as statin side effect. However, there's not many studies on this SNP association with statin efficacy. There is a previous in vivo study using GATM deficient mice has shown that significant decrease can be seen in the GATM deficient mice in serum cholesterol levels as compared to the wild type, but this finding is yet to be replicated in human study. Our current study has shown that GATM RS9806699 was not significantly associated with any lipid parameters, therefore doesn't affect the statin efficacy in our study population. Therefore, as a conclusion, to answer our problem statement, whether are there any association in between these genetic polymorphisms and statin efficacy in reducing lipid level? Yes, statin efficacy is associated with genetic polymorphisms, in which we found out that ApoA5 RS662799 carriers had exerted a better response than their non-carrier counterpart. While for our second SNP, GATM RS9806699 carriers, however, was not associated with statin efficacy. For future studies, our studies had limitations, and therefore we suggest a future multi-center study involving more SNPs and a stricter recruitment criteria to increase the understanding in this statin pharmacogenomics field. Here's a list of references. Thank you.
Okay, thank you, Amir Faiz. Let's welcome you for 5 minutes Q&A session.
Amirul. Ya, Assalamualaikum Amirul. Tak dengar. Tak dengar Amirul. Ha. Okey Amirul.
Hello?
Okey dah.
Okay.
Amirul okey dah. Amirul ke Faiz?
Faiz boleh?
Okay, Faiz. Baik, Faiz. Is it the statin is a drug of choice for the treatment?
Yes.
Is it?
Yes.
Yeah, okay.
Yeah.
It's looking at that thing is that seems to be macam you are macam towards kepada personalized medicine ke?
More like that. Yes. Yeah.
Is it because you kata you new conclusion tu lebih kepada genetic polymorphism, kan? Meaning that the presence of the genes or certain characteristic of the genetics material, then the statin is will be suitable. Am I right to say that?
Yeah. That's my point.
And then that is the reason, whatever it is. So otherwise, statin ni kalau berdasarkan pembacaan saya lebih kurang whatever it is, a lot of side effect kan dia punya drugs. It's not good kan?
Yeah. Yeah. A common side effect is the muscle pain lah. Statin induced muscle pain or myopathy.
And then leading towards that, why tak study ni is that macam SNP ni macam kind of very personalized thing whatever it is. Ada kind of you reading ataupun the strategy of the design kan kepada high throughput punya screening.
High throughput means doktor nak increase the sample size.
And then more genes is being screened, something like that.
Okay. So doktor punya question boleh ulang balik? I mean question ulang balik. Saya tak clear.
Something like that macam kan sometimes is that while kalau nak working on the one gene, the conclusion tu macam lambat, nothing whatever.
Betul.
And then the start, a lot of things cannot be accessible. Kita agak terdelay ataupun lambat research kita whatever it is. And then that's why is mungkin lah maybe you know better. It's about the high throughput screening, to make it things ataupun lebih robust punya study design, something like that.
Okay. For me, that is one of the limitation of my study I made because we focus on one gene, one SNP only. So we focus a gene interaction or other confounding factors in our study. Just focus on one factors only. So that I admit is my limitation. So that's meant for more SNPs and more stricter criteria for the next study. So that's my opinion for the question.
Okay, quite interesting. Okay, Dr. Yus, please, Dr. Yus, continue.
Assalamualaikum Faiz.
Assalamualaikum.
Actually, I may be missed some point of your presentation just now, but based on your study, did you find a relationship between these two genes? Because you mentioned APO-A5 is actually increased risk for obesity, and then ATM is actually for the statin.
Okay, Dr. Slow. Tak dengar. Sorry.
Okay. Based on your study, did you find any interrelationship between these two genes? Like for example, if let's say one gene is suppressed and the other one would be enhanced. Because you mentioned that APO-A5 is actually to increase the risk of obesity and the ATM is actually for the statin induced myopathy, right?
Okay.
Interrelationship between these two genes.
Oh, okay. So your question is there any interrelationship between these two genes? Okay. For that, we didn't focus on gene interaction. We just focus on one SNP only. So we just find the association between each SNPs. Okay. So not between them.
So you just study the right.
Terputus. Suara detik.
Terputus.
And
Dia betul-betul tak tahu? Betul-betul.
Suara Dr. Yus terputus.
Betul-betul. Dia betul-betul.
okay, let's just study. Yeah.
Can you type? Can you type?
It's okay. So meaning that you just study the expression of these two genes separately, right?
Okay, yes, yes.
Okay, that's all.
Okay. Is there any
That's all.
Oh,
Thank you. That's all. Thank you,
Is
Faiz.
there any more question?
thank you, Doctor.
No. Okay. Alhamdulillah. It's lucky that we had within our 5 minutes. Okay. Without further any delay, call upon our next presenter with the code M37, Nishantini, to present her research entitled Annexin A1 mediated modulations in clean and cancerous mutants treated breast cancer cell line. Please welcome.
A very good day to everyone. I'm Nishantini Perantan, a PhD student from University of Bangsa, Malaysia. Today, it's my great pleasure to share with you our study entitled Annexin A1 mediated modulation on clean cancer mutants treated breast cancer cells. The era of personalized medicine is likely to see an escalation in the use of biomarkers. The discovery of biomarkers in cancer drastically changed the course of cancer treatment. One such potential biomarker that we will explore is annexin A1, a well-known immunomodulatory protein where it modulates innate and adaptive immune responses. In recent years, there has been an exponential interest in the role of annexin A1 in cancer due to the increasing number of studies investigating the effect of this endogenous protein in different types of cancer. Indeed, increased level of annexin A1 expression has been reported in pancreatic, hepatic, stomach, skin squamous cell carcinoma, as well as colon cancer. However, the expression of annexin A1 in breast cancer is more complicated with contradictory reports. As we all know, breast cancer is a heterogeneous disease with a high degree of diversity present among individual patients as well as between and within tumors. Therefore, breast cancer calls an urgent need to the discovery of more and more biomarkers. Higher expression of annexin A1 is associated with basal-like breast cancer, particularly within the subgroups that are used to define poor prognosis, such as basal-like breast cancer, specifically triple-negative breast cancer, compared to other subtypes of breast cancer. Increased expression of annexin A1 in cancer is often related to its participation and regulatory role in cancer cell invasion, migration, proliferation, and epithelial mesenchymal transition. And the diagram here shows the potential influence of annexin A1 on macrophages that is required for and played an important role in the tumor microenvironment. So this annexin A1 macrophages is actually required for alternatively activated M2 macrophage polarization, which promotes the tumor growth and also the invasion. Given the importance of annexin A1 in modulating the immune responses, it is surprising that its involvement in the process of triple-negative breast cancer immunomodulation has not yet been fully explored. Immunomodulation by plant-derived products is of active interest in therapies being designed to combat cancer. Clinacanthus mutans, locally known as Bulalai Gajah, in English it is called Sabah snake grass. It also is one of the important species from Acanthaceae family and has been used as vital herbal medicines in tropical Asia. Interestingly, it has gained much attention due to its immunomodulatory and anti-cancer effect. However, mechanism remains unclear. Thus, our study aims to investigate the association between annexin A1 expression in breast cancer and its role following treatment with clinacanthus mutants. This will be achieved by specific objectives. Number one, to characterize the annexin A1 expression in MD-MB231 and NCF7 cell line by comparative transcriptomic analysis. Second, to determine the enriched pathways of the annexin A1 associated differentially expressed genes, DEGs. And third, to investigate the cytotoxicity of clinacanthus mutants treatment in MD-MB231 and NCF7 cell line. Here we have our experimental design. We use two different cell lines, which is MD-MB231 bearing TNBC characteristics, NCF7 the non-TNBC cell line. We further collected the cells to do RNA extraction, which will be further used for RNA sequencing analysis. We managed to do gene expression quantification and differential expression analysis, as well as functional analysis. We also validated the annexin A1 expression by using real-time PCR. And then we subjected these two cell lines to do SRB assay to evaluate the cytotoxicity of clinacanthus mutants treatment. And then all the results obtained were statistically analyzed using SPSS. Let's move on to results and discussion. Figure 1 is the volcano plot of the transcriptomics profiles of MD-MB231 versus MCF7. Compared to two cell lines, we found 4,100 upregulated genes and 4,547 downregulated differentially expressed genes, DEGs. So to prioritize only the annexin A1 related functions, we further filtered the DEGs and we successfully identified 461 annexin A1 associated DEGs, with 286 genes upregulated and also 175 genes downregulated. So to aid the filtering process, we identified the overlapping DEGs with a public accessible RNA sequencing dataset which profiled the transcriptomics of wild type and annexin A1 knockout A549 cells. Figure 3 and Figure 4 shows the heat maps based on RNA sequencing analysis. Generally, the red color indicates upregulation, whereby the blue color indicates downregulation. So, based on these results, annexin A1 and also FPR1 and FPR2, the only known receptors of annexin A1, are found to be highly expressed. So, the heat maps of the annexin A1 gene signatures clearly showed that the annexin A1 and its interacting genes were upregulated in MD-MB231 cells, the high annexin A1 expressor. In view of the pronounced effect of annexin A1 on oncoimmunity, we questioned if annexin A1 orchestrates the cell fate of macrophages to favor either pro or anti-inflammatory immunity in tumor microenvironment. So we therefore interrogated the expression trend of various mediators related to tumor-associated macrophages, mainly tumor necrosis factor alpha, interleukin-1 alpha, interleukin-1 beta, and also TGF beta. So in high annexin A1 expressors like MD-MB231, the M1 and M2 gene signatures were overly expressed, suggesting a heightened macrophages activity compared to low annexin A1 expressor, which is the MCF7 cell line. Moving on to the top 20 enriched functions of the annexin A1 associated DEGs to answer objective number 2. Here, the circle here, different size of circle indicates a different percentage of enrichment, and the different colors in a circle indicates different p-value. Interestingly, immune-related activities, including innate and adaptive immunity, were overrepresented, implying a crucial role of annexin A1 in the modulation of tumor immune landscape. Also, pedokine signaling by cytokines and chemokines, tumor microenvironment-related pathways, as well as metastatic activities like HIF1 alpha signaling and regulation of matrix metalloproteases were also well implicated. To unveil the relationship between different enriched functions from annexin A1 associated gene, we used the output from gene set enrichment analysis to construct a connectivity network. Again, the red dot here indicates upregulation and the blue dot indicates downregulations of the EG. So according to the network constructed, annexin A1 has a predominant modulatory effect on immune cell response, cytokine and chemokine activation, and also macrophage activation were highly intertwined. So importantly, this constructed network provides novel insights on the role of triple-negative breast cancer annexin A1 in macrophages activation and also toll-like receptor signaling, which has not been reported so far. Figure 1 shows the real-time PCR analysis of the validation of annexin A1 expression in 3 different breast cancer cell lines, MCF7, the non-TNBC cell line, and MD-MB231 and also MD-MB468, the triple-negative breast cancer cell line. Here, clearly, MD-MB231 and also MD-MB468, the TNBC cell line, expresses high level of annexin A1 compared to the non-TNBC cell line. We then conducted SRV assay to evaluate cytotoxicity of clinacanthus mutants on MD-MB231 cells and MCF7 cells, which answers objective number 3. So here we can see that clinacanthus mutants ethanolic extract showed significant cytotoxic effect on MCF7 cells with IC50 of 0.85 mg per ml after 48 hours and also 72 hours of treatment, whereby MD-MB231 did not show any significant cytotoxic effect even up to 72 hours. Taken together, based on the transcriptomic profiles, annexin A1 may exert a profound regulatory role in oncoimmunity, tumor stromal crosstalk, and epithelial mesenchymal transition in the tumor microenvironment. Similarly to what was observed in other cancer subtypes, annexin A1 expression in triple-negative breast cancer cell line is associated with aggressive cellular behavior and resistance to many treatments. So the disparity in Klinekanthus-Newton's treatment responsiveness of breast cancer may be linked to the increased annexin A1 expression in triple-negative breast cancer and not in the non-triple-negative breast cancer. So as a result, the immunomodulation mediated by annexin A1 in Klinekanthus-Newton's warrants further evaluation to understand the downstream mechanisms of immunomodulation. Over 1.6 million diagnoses every year. One woman dies from breast cancer every minute. Male breast cancer accounts for 1% of all breast cancers. Together, we invest in hope for all women and their loved ones. Research brings us one step closer to achieving our goal, which is to develop new effective and targeted treatments. Thank you.
Okay, thank you, presenters. Let's proceed with the Q&A session.
Mana? Who is the presenter? Okay, Lishanti.
Yeah, I'm the presenter. Yes, Dr.
Okay,
Lishanti.
very extensive
Yeah.
works. You're doing PhD, I think, I guess. Is it?
Yeah,
You're doing
yes, Dr.
PhD.
I'm doing PhD. Yeah.
I think okay. This is the in silico data, kind of, isn't it? In silico.
Transcriptomic profilings of cell-based
Kind of in silico
next gen,
thing, whatever it is.
next generation.
You're predicting whatever it is. So what's next?
Yeah, so now this transcriptomic profilings is basically a fishing process to identify the annexin in A1 mediated modulation. So from here, we understand that this annexin A1 triple negative breast cancer has a very a very good role in tumor microenvironment. So in our further studies, we are planning to do a co-culture study to mimic the tumor microenvironment, which means we will grow together our triple negative breast cancer with macrophages. So we will mimic a tumor microenvironment and we will look at the interactions between this triple negative breast cancer and macrophages. And also we will do some knockdown studies on annexin A1. So we will silence this annexin A1 and we will further see what the silencing can do. And we will also further treat it with Klinekanthus nutans, whether it can react back after silencing with annexin A1 knockdown.
Okay, good. So just to look at it, because the spectrum is so broad thing, whatever it is, you can see that physiological effect thing. That's why I'm just to know is any streamline, area that you would like to see that you see is what is the most important for the breast cancer, I guess. Yeah, that is
exactly, doctor. Yeah, because this is more likely it is going to with tumor microenvironment. So we have to mimic that environment in vitro settings by co-culture.
I agree with you. Yes. Dr. Yus?
Thank you, doctor.
Hi, I'm Shani, is it,
Hello, doctor.
May I know the Klinekanthus that you use? What types of extract did you use? Fruit extract? What kind of?
The extraction.
Yes, crude extract, doctor.
Yeah, organic or inorganic?
Yeah, crude extract.
Is it organic or
Pardon, doctor? Sorry?
Is it organic or inorganic extract?
Yeah, ethanolic extract.
Ethanolic.
Ethanolic extract. Okay. Maybe I need to use them now. And the data you show previously, the mapping of the downregulation and upregulation of the annexin and the EG is actually before treated with the extract, right?
Yeah, exactly, doctor, before treating with extract.
Okay, so after this, then you will proceed with the extract by using glucose culture.
Yeah, that is what we are planning to do, doctor. So to fully understand the concept.
That's good. Okay, thank you.
Thank you so much, doctor. Any more questions?
I know.
Are you comparing the different type of extractions, or just we're going focus on ethanolic only?
Yeah, actually, this is a continuation of a previous study. So they have done like with different extracts, but ethanolic extract showed a potent effect on this cell line. So we further refined to this ethanolic extract.
Okay, so it's already been conducted, and then you focus on it. Okay.
Yeah, has been conducted.
Okay, thank you.
Thank you, Doctor.
Well done, presenters. For the next, I shall pass the baton to Mulu Levelan Lesta with the code M44 to present her research entitled Molecular Detections and Antimicrobial Resistance Profiles of Extended Spectrum Beta-lactamase producing Escherichia coli in broiler chicken farms in Kota Baru, Kelantan. Please proceed.
Hello everyone, my name is Mulu Lemlem and I'm going to present on the molecular detection and antimicrobial resistance profile of extended spectrum beta-lactamase producing E. coli in broiler chicken farms in Kota Baru, Kelantan. These are the co-authors. I'm under supervision of Dr. Erkion Aklilu. I'm from Faculty of Veterinary Medicine, University Malaysia, Kelantan. These are my presentation outlines. It starts with the introduction. Antimicrobial resistance is one of the most serious global public health threat. And currently about 700,000 people are dying because of this antimicrobial resistance each year. This number is expected to reach to 10 million deaths each year by 2050 if any corrective action is done on this issue. Antimicrobial resistance threat have reached in its alarming level because of the limited options of treatment for the multi-drug resistance antimicrobes, especially antimicrobial multi-drug resistance bacterial infections. The main drivers of antimicrobial resistance is misuse and excessive use of antimicrobials in human and animals. The excessive use of antimicrobial in veterinary medicine, food animal production, and agriculture results in the emergence of antimicrobial resistance. Food producing animals are considered to be an important reservoirs of various multi-drug resistance microbes. Animal originated Enterobacteria, particularly Escherichia coli, can be pathogenic to human and it may transfer a resistance gene to other pathogens. Enterobacteria exhibits antimicrobial resistance mainly due to the enzyme production. We call it extended spectrum beta-lactamase enzymes. And these Enterobacteria are more prone to carry resistance gene. As we all know, E. coli is a harmless facultative anaerobic bacteria, which is most commonly found in the GIT of human and animals. However, it is also a potential pathogenic bacteria that can cause variety of diseases in human and animals. And it is also a major cause of mortality and morbidity in poultry farms. Limited research have been done in Peninsular Malaysia, but there is no data on the prevalence of ASPL producing E. coli and its encoding genes from broiler chickens in chicken farms in Kota Baru, Kelantan. Therefore, this study was intended to fill this gap. Methods and materials, a total of 121 presumptive E. coli were isolated from cloacal swab collected from broiler chicken farm in Kota Baru and nearby districts in Kelantan. The E. coli isolated taken from stock culture were streaked on Makonki agar. Lactose fermenter colonies from Makonki were subcultured to EMB agar and incubated at 37 degrees centigrade for 24 hours. The green metallic sheen colonies were considered to be an E. coli, and these colonies were further tested for their biochemical tests. The biochemical characteristics that was checked were TSI, citrate, urea, indole, methylene red, and motility. Isolates were further confirmed by PCR that targets for 16S rRNA using species-specific primer for E. coli. Antimicrobial susceptibility test was done using Kerry-Blair disk diffusion method. Bacterial suspension was prepared equivalent to 0.5 McFarland standard, and it was lowered uniformly over the surface of Northampton agar plate using sterile cotton swab. antibiotic susceptibility profile of 14 antibiotics, different class antibiotics that are clinically relevant to human and animals were tested. Zone of inhibition were interpreted based on the CLSI recommendation. Those are the antibiotics that were used for the antimicrobial susceptibility test of the isolated E. coli. For the PCR confirmation of E. coli, DNA was extracted using bacterial DNA extraction kit. This extracted DNA was amplified using species-specific primer, the 4 primer. PCR product was analyzed using agarose gel electrophoresis. Gel imaging was done using gel documentation system. ATCC 25922 was used as a positive quality control. To detect SPL encoding genes, extracted genomic DNA was amplified with specific primers, and the BLA CTX and BLA TEM genes were detected. PCR product was analyzed using agarose gel electrophoresis for the identification of SPL encoding genes. These are the forward and reverse primer sequences of the primers that are used in this study. Result and discussion in this study, 121 presumptive E. coli were isolated from cloacal swab by routine bacteriological and biochemical characteristics. Out of these, 80% of them were confirmed as E. coli by PCR using species-specific primers. And this image shows that the samples that are positive for the 4 primer. Antimicrobial susceptibility profile. In this study, E. coli isolates had relatively higher resistance to amoxicillin, tetracycline, and sulfametoxazole trimetoprine, whereas colistin shows the lowest resistance. Similarly, higher resistance rate of tetracycline and sulfametoxazole trimetoprine, which is 91.4 and 74.2 relatively, were reported from previous study on broiler chicken farms in Malaysia. In the present study, E. coli isolates were resistant to carbapenem antibiotics, meropenem 28.9 and ipenem 17.5%. Overall, 93.8% of the isolates were resistant to at least 3 of the 14 different classes of antibiotics. This shows that most of the E. coli isolates were multi-drug resistance. Excessive use of this common antimicrobials in poultry production has been reported to cause multi-drug resistance. As you can see from this table, this shows the percentage of resistance of isolated E. coli to different antibiotics. And the higher resistance was shown for amoxicillin, tetracycline, and sulfametoxazole, and the lowest resistance was shown in colistin. The SPL encoding genes in E. coli. In this study, BLA-CTXM 62.9% and BLA-TEM 45.4% of SPL encoding genes were found. This BLA-CTXM 62.9% were slightly lower than the previous study from Iran, which is 60.3%, and it is also lower than the previous study from Malaysia, that is 100%. And the BLA-TEM 45.4% were higher than the study from Iran, which is 37.7, which is chicken origin, but slightly lower than published data from the surface water isolates of Enterobacteria in Malaysia, which is 47.4. Similarly, it has been reported that higher prevalence of BLA-CTXM followed by BLA-TEM harboring E. coli were isolated from human clinical samples. In addition, 16.5% were positive for colistin resistance encoding genes MCR1. The previous studies from Malaysia shows that high prevalence of SPL as well as colistin resistance E. coli were found from raw chicken meat. And majority of the SPL gene encoding gene in this study were found to be BLA-CTXM gene, which is similar to many published studies in broilers conducted globally. This table shows the distribution of ASBL encoding gene in E. coli. Our study shows that 22.7% of E. coli isolates were harboring both CTXM and BLA-TEM genes, which is slightly lower than the previous study from Philippines, 33.3%, while 16.5% of the isolates were positive for both MCR1 and SBL genes. Similar finding also reported from studies in Egypt, Nepal, and China. 6 of the isolates were found encoding both the BLA-CTXM, BLA-TEM, and MCR1 gene. This table shows the coexistence of ASBL genes with MCR and the coexistence of both BLA-CTXM and BLA-TEM. Overall, 84.5% of the isolated E. coli were positive for at least one ASBL genes. This finding suggests that ASBL genes in E. coli isolates were distributed in broiler farms. The spread of ASBL producing E. coli isolates from apparently healthy food in animal indicates that this commensal E. coli may serve as a resistance gene reservoir, which have a potential risk of transfer to humans through food chain. Conclusion. This study showed that a high prevalence of ASBL genes in E. coli isolated from broiler chicken farms in East Coast Malaysia. This provides insight that ASBL producing E. coli is spreading among the broilers in the farm that have a possibility of dissemination of this resistance bacteria to human via food chain. Our finding shows that BLA-CTXM is the most prevalent ASBL producing gene. Most of E. coli isolates were multi-drug resistance, which may have a potential risk of ASBL producing E. coli infections in animals as well as in. These are some of the references that I used. Thank you for your attention.
Okay, thank you, presenters. Let's welcome for 5 minutes Q&A session. You can now switch on your microphone and camera.
Hi, Lulu.
Hi, doctor.
You are from? You are from?
I'm from Ethiopia.
From Egypt. You are from
Ethiopia.
Egypt. Okay. From Ethiopia.
Yes.
Habsha.
Yes.
Okay. Okay, Lulu. Can I call you Lulu or you want
Mulu.
me? Mulu.
Yes.
Mulu. When you are looking for the BLA gene, are you actually looking for the ampicillin resistance, or this BLA gene also associated with another or other antibiotics?
Third generation cephalosporin. I'm looking mostly. I'm focusing on the third generation cephalosporin.
Cephalosporin, is it related to the
ampicillin? Not ampicillin. Third generation, especially for the cefotaxime and ceftazidime.
And then this BLA gene is actually we actually looking for the beta lactamase resistance mechanism?
Yes. Beta lactamase, yes, the beta lactamase resistance. It code a gene that called BLA CTX, BLA SHB, and BLA TEM.
Yeah, that is my confusing me. Something normally in a beta lactamase is only normally related to the ampicillin resistance, something like that. Okay, I'm not.
It is also related to the third generation cephalosporin drugs.
I see. All right, whatever it is. Generally, can you the mechanism of that cephalosporin resistance?
Yes, the resistance mechanism of the SPL gene is due to the production of enzyme that hydrolyzes what you call the beta lactam ring of the bacteria.
Okay. All right. And then one last question. Why MCR1 you're looking for as well? Why the MCR gene also you're looking? Any associated with that thing that, or you are predicting something?
No, just what I mean is here, why I include here MCR1 is because it was found binding together with the ASBL gene. That's why.
Is it?
Yes.
So there is a relationship lah between that MCR1 and also the.
No. They are two different because MCR is for the colistin resistance.
That's why I'm
Yeah.
looking that where
No,
is
no.
connect these two things is I'm wondering.
No, just to express their coexistence. No,
Right.
there is no relation between this.
Whatever it is. Okay, thank you, Dr. Yus. Please, Dr. Yus.
I have no questions. No question from me.
Okay, doctor.
No question. No question from Dr. Yus. Yes,
Okay.
okay. So can I ask you one question?
Okay.
Then, do you think that One Health will help in this genetics in this antibiotic resistant problems? One Health approach.
One
Health approach.
One Health.
Huh?
I don't.
You don't understand. No? You don't include
Yeah.
that also because I found that your supervisor they are One Health a strong whole people in diabetes. Okay, no. Okay, diabetes. Okay, never mind. Okay, that's all from me.
Okay. Thank you, doctor.
You doing PhD or master?
I'm PhD. I'm PhD student. This is one of the project.
Project, no?
Mm.
And then if I got time, Shamimi, got time?
Yes, we have 2 minutes.
2 minutes, okay. If you can compare, Lulu, is just try to learn from you the practices in Egypt, if you're not sensitive now, with the Malaysia thing, whatever it is it similar or in Egypt is better management of the broiler on the antibiotic resistance?
Actually, I don't know I am from Ethiopia.
Oh, Ethiopia, in Ethiopia.
Yes.
Is it no broiler farms there, or there is a Maudi?
Yeah, there are broiler farms, I think, but they are not as large as like Malaysia.
So the antibiotic
most of
the controllable.
Yeah. Yes, most of them are also the village, like what you call like village farms, not like a big farm like this.
Kind of the backyard farm, something like that.
Yes. Most of them are like that. But use of antibiotics is also there. It is also the concern also there also they have a concern of antibiotic usage.
Because we are in Malaysia, we are so much scared with this antibiotic resistance problem thing like this. Yeah, we are really scared.
Sure.
Okay.
Sure.
Okay. All right, Shamimi, I pass to you, Shamimi. Thank you very much.
Thank you.
Okay, thank you, judges and welcome presenters. Okay, let's have our last presenter, Nur Alisa Kamarudin with the code M45 to present her research entitled Therapeutic Potential of Neural Stem Cell Preconditioned with Gelatin Enriched Fraction for Ischemic Stroke Treatment. Please welcome.
Assalamu alaikum and hi, my name is No Alisa binti Kamarudin from University Science Malaysia and my code participant given is M45. I would like to present to you my study entitled Therapeutic Potential of Neural Stem Cell Preconditioned with Gelatin Enriched Fraction GEF for Ischemic Stroke Treatment. Introduction. What is stroke? According to statistical analysis in 2020, there are 5 major causes of death in Malaysia, and cerebrovascular disease such as stroke been placed as top 3 major causes of death in Malaysia. The stroke have been divided into 2 major groups, which are ischemic and hemorrhagic. In my study, I want to focus on ischemic stroke since it's been reported most of cases of stroke is ischemic. What is ischemic stroke? Ischemic stroke is due to the blockage of a blood vessel by foreign mass that we call as embolus, which block the blood flow to the brain area, and this lack of blood flow supply causes a brain self-death and it caused a disease called as stroke. There are many methods to establish ischemic stroke model, but in this study, we choose 81 model, which we use vasoconstrictor protein or endothelin-1 to constrict the blood vessel in the specific area. The source of treatment we used in this study is Quercus infectoria, or we call it as Manjukani in Malaysia. It's commonly found in a small shrub found in Greece, Asia Minor, and Iran. It's been reported have a potential as anti-inflammatory, antiviral, antidiabetic, larvicidal, and etc. Galantamine was found abundantly in Manjukani. Galantamine or tannic acid are commonly used in many studies to evaluate its potential, such as antioxidant activity, anti-inflammatory effects, antimicrobial effects, anti-carcinogenic activity, and neuroprotective effects. So we want to enhance its potential on neurostem cell to treat ischemic stroke. The problem statement and the objective of this study. As we know, stroke causes irreversible damage, but with the advance of technology, it could be treated using endo and exogenous neurogenesis technology. However, due to low survival rate and low turnover rate, exogenous transplantation is still not efficient to regenerate that cell. So, we tried to enhance the therapeutic potential of a neural stem cell using galantamine of Quercus infectoria. The objective of study, we want to optimize the effective concentration of galantamine-enriched fractions of Quercus infectoria and to determine the effective concentrations of GEF on neural stem cell, and we want to identify the neurological effect of GEF on established rat stroke model. Here are my general methodology of my study. We extract and characterize the plant, recondition the neural stem cell, and we transplant the neural stem cell into ischemic stroke model. Here are the results obtained from this study. After being extracted using chocolate for 6 hours, the plant sample being fractioned into 6 fractionations. The yield productions of fractionation show the effort given the highest yield than the other fraction. We did a Hplc analysis to check the presence of galantamine compound for each fraction, and we found the galantamine were appeared clearly in F1 till F4. However, we still proceed to Hplc analysis to quantify the galantamine for each fractionation samples, and we found F4 showed the highest galantamine content, and we decided to use it in our study as galantamine enriched fraction or GEF. We proceed to DPPH assay to evaluate the antioxidant potential of galantamine compared to standard and the crude itself, and it shows significantly higher antioxidants at 20 microgram per ml compared to another. We preconditioned the neural stem cell using GEF, and we did a cytotoxicity assay, and we found the proliferation rate of neural stem cell treated with 12.5 microgram per ml GEF was significantly highest after 24 hours. This is the view of cell after being treated with 12.5 microgram per ml GEF. We observed the behavior of rat ischemic stroke model for 2 weeks, and we did neurological deficit score, which are detections of abnormal posture, hemiplegia, and limb dysfunction detection. We observed that the treated with GEF preconditioned neural stem cell group showed improvement better than the other group, especially at day 2. So what I can conclude in this study is the GEF demonstrate significant higher radical scavenging activity at 20 microgram per ml, and it showed that GEF significantly improved the NSC growth rate after 24 hours at 12.5 microgram per ml. And the GEF preconditioned neural stem cell treated stroke rate shows significantly reduced of stroke severity, and it could be a promising novel therapeutic agent in enhancing NSC therapeutic potential for ischemic stroke therapy. References. Thank you.
Thank you, presenter. Let's welcome Q&A session.
Okay dah. Dr. Yus tanya dulu kan? Dr. Yus pakar benda ni.
Sudah.
Hi. The problem, maybe I can answer. Actually, based on your result just now that is presented, you use a few different dosage for the preparation assay, right, for NSC. But then you choose 12.5, right, if I'm not mistaken. If you look at your graph, there is no different changes, like for the growth rate of NSC after you treated with the higher dose. Do you have any explanation on that?
Thank you, Dr. Yus. This is from my MTT essay result, right?
Your proliferation.
Proliferation, MTT essay. Okay, actually, during we tried, when we decided to use dosage 12.5 microgram per ml at 24 hours treatment, actually it's been supported with other article also that many preconditioned method using 24 hours as treatment to NSC. In addition, my result from the number of cell viable are most viable at that concentration of 12.5 microgram per ml.
If I'm not mistaken, when you use the higher dose, the form is actually not significantly it maintains at the viable rate, right? At 4.5. So it means that and at the higher concentration over than 12.5 microgram per ml, there is no effect with the glycemic. Higher than that, there is no effect.
The toxicity effect on my neural stem cell. That's why we want to use minimize as we can now to avoid toxicity effect.
Yeah, but in your result, just so you display your result, at the higher dosage, actually there is no effect, right, for your neural stem cell.
Actually, there is effect. The viable become decrease when we increase the dosage concentration. If you can see in my graph lah.
For your toxicity, I saw that there is no effect on the inhibition code. Right, okay. Maybe I just
Okay. Thank you,
that.
Doctor.
Okay, just another one question.
Okay.
For study for the so it's actually still ongoing.
Sorry, can you repeat the question again?
The ischemic stroke, effect of this NSC on ischemic stroke. How did the progress of this
Okay. Okay. Part animal. We already done that part. Okay. Okay. Currently, we are doing histology part. HNE and TTC analysis.
so it's not included in the presentation just now, right?
Yes.
Did you find there is retain of the donor stem cell of the process into the ischemic frame? Is there any limitation on that procedure?
Uh, the limit. Sorry, doctor. Can you repeat the question again?
When you
Maybe the connection's not good.
sorry,
Please, sorry.
you cannot hear me clearly, right? Can you hear me clearly?
Okay. I can hear you.
When you are grafted into the ischemic brain, is
Okay.
there a limitation on that procedure?
Okay, the limitation of my procedure is during surgery, which I want to plant the guide cannula on the head of rat. We found that when the rat is not recovery enough, the rat become dying and die. That part is quite complicated for us to maintain the number of samples.
Okay, thank you, Dr. Adam.
Okay.
Saya akan pass to Prof.
Cukup tak ada. No question. Saya dah follow. Okay. Thank you very much. Cepatlah. Dr. Yus punya soalan pun fine. Tapi Alisa, it's that saya normally lah kita tanya kalau when we do the extractions, dia ada different extraction kan, different type of extraction. Have you tried everything? Dah optimize the extraction lah.
Kami buat untuk decide nak teruskan dengan extraction tu, kami dah optimize dengan dua jenis solvent, iaitu ethanol dengan air. Dengan distilled water. Distilled water. Yeah.
So ethanol better?
Air, distilled water, aqueous punya extraction lagi better. Yeah.
That's right. Back to Shamimi.
Okay, well done presenters. Thank you judges. So we have come to the end of our oral sessions and we finished earlier, 30 minutes compared to yesterday. So thank you to all presenters for your participation and cooperations. And this is quite spontaneous. Since we finished earlier, can I ask our judges can we have a general advice from the judges to our future scholars? Maybe some wise word from you guys, from both Dr. Ridwan and Dr. Yusmazura. Maybe a short and simple wise word. Because we know the conference is the only way for us to meet the expert directly, right? So why not we didn't let this chance go just like that.
I think
Sorry, this is spontaneous from me.
Saya pun voluntary. It's just a voluntary my observation throughout this. And then because of the time limit lah. Most of the time because the time limit Shamimi ataupun it's not because of student limitation or the student study whatever it is. And I believe whatever all the studies so far within for these 2 days punya I follow your conference. I found that it's good study, good student and good presentation and so on. Only the time is so limit. And then I noticed that the angles ataupun kepuasan satisfactory for us as a judge pun to ask a question also, is very limit. And then you Shamimi is very strict with the time thing, whatever it is. And then whatever it is, it's good anyway. It's a time thing is very good. But the point is that it's also what you call it, the student when they are presenting, they also would like to be asked so many questions so that they can answer a lot of things that they're not able to present within that 10 minutes given. Betul tak, Syami? So that's the thing. So I think this is whatever what you call it, this conference actually is a train how within this 10 minutes, is it for us to actually polish kita punya skill, polish kita punya scholarly critical thinking, how to deliver that thing effectively. Okay, I believe that all of us can learn from each other. So something like that. Oh, like this. We can tackle things like this. And then in the future, I believe, Shamimi, that all of us will be improved definitely, inshallah. That is my things lah. My comment lah. Brief comment, something like that.
Regarding the times, actually we have Mr. Amirul Faiz, the chairperson for our conference. So Amirul, can you comment about the time allocation? Because I'm only following the orders. Amirul?
We'll better next event in terms of time allocation. So I can
Because
explain to.
I quite agree with Dr. Ridwan because I think the Q&A session is actually a learning session. It's a time for us to learn. Okay. Dr. Yusmazura, you have some words? Or maybe.
Thank you, Shamini. First of all, congratulations to all the presenters. Actually, very interesting research that presented. I also learned a lot from all of you. And I also agree with Prof. Ridwan, where there's a limitation due to time, so that we also cannot go into the research in detail. Like for the example, the methodology is so brief so that I cannot catch up actually what is actually you're all exactly doing anything to relate the results. So that maybe in future maybe we can see into that.
Okay.
Thank you.
So thank you, judges, for willing for this spontaneous questions. Okay. To all, before we end the sessions, may I ask to all participants and everyone, and to all non-presenters as well, to switch on your microphone and to switch on your camera, to have, or we have a visual session or photography session. So, Mak Syari, our yesterday presenters, please, you can also switch on your camera. Adisa and Mulu Lamlam. Yeah, our PIC, are you ready?
Yep, I'll start taking pictures at the count of 3, all right? So 3, 2, 1, smile. One more. 3, 2, 1, smile. All right, thank you, everyone. Good job.
Okay, thank you, everyone. So before you leave this virtual room, I would like to be reminded for you to fill in the feedback forms. That fill in the feedback form via the links provided in the chat box. Okay, thank you. Thank you, everyone, and good luck.
Thank you very much. Okay, baik. Assalamualaikum.
Assalamualaikum.
Thank you, Doctor.